Today we will proceed from polysaccharides. What is polysaccharide? More than 10 monosaccharides are joined by glycosidic bond. Okay, so it is also colloidal in nature. What are the different types of polysaccharides we know about? Homopolysaccharide and heteropolysaccharides. In homopolysaccharide, all the monosaccharide units are same, whereas in heteropolysaccharide, the monosaccharide units may differ. Okay. So the examples are as such. Homopolysaccharides, they can be divided into plant origin and animal origin. In plant origin, we have starch, cellulose, inulin, also chitin and dextran. They are not from plant origin. Animal, they are from bacteria and other crustacean group animals, okay, insects. In the glycogen, it is found in animals, and it is a storage form of animal carbohydrate, whereas the storage form of plant carbohydrate is starch. Cellulose is the structural component of plant. Now coming to the heteropolysaccharides, it can also be divided into two groups, plant and animal origin. So in plant origin, we have agar, agarose, gum, pectin, hemicellulose, all these things. And in animal origin, mucopolysaccharides or glycosaminoglycans. Okay? Okay. So homopolysaccharides, now coming to individual homopolysaccharides, that is starch. It is the reserve carbohydrate in plants. What is the what are the sources? Mainly all the starch and cereals and vegetables containing starch like potatoes and cereals. Okay. It is composed of amylose and amylopectin. What is amylose? Amylose is the straight chain of, what is this structure? Alpha or beta? Alpha. So this is alpha D glucose joined by alpha 1,4 glycosidic bond. So amylose is nothing but straight chain of alpha D glucose joined by alpha 1,4 glycosidic bond. Whereas amylopectin is a branched form of glucose polymer. Whereas the 1,4 connections are there, but the branching starts at 1,6 glycosidic bond. So starch is a composition of both amylose and amylopectin. Okay. So the amylopectin makes the insoluble part. Okay. Amylopectin forms the gel-like structure when it is mixed with water, and the branches are at every 20 to 25 glucose unit. So in a long chain, after 20 to 25 glucose units, one branch will be there. Okay. Next, for identification of starch, what is the test we do? It is iodine test. What happens if we mix starch with iodine? It will give a blue color, and on heating the color disappears. On cooling, again the blue color reappears. Okay, but starch is non-reducing. Why? Because the free sugar groups are, or the free functional groups are not that much. Okay, but if we hydrolyze the starch, and starch is hydrolyzed by nothing but amylase enzyme. Okay, so how many amylase enzymes are there in our body? Amylase, salivary amylase and pancreatic amylase. So the hydrolysis of starch starts from our oral cavity. The moment we start eating starchy food, our salivary amylase starts hydrolyzing the starch. আচ্ছা এটা হচ্ছে না, এটাকে একটু চেঞ্জ করতে হবে মাঝে মাঝে। So when we start hydrolyzing of starch, what are the products we get? Dextrins. Okay, so what is dextrin? Dextrin is a product of hydrolysis from starch. Smaller units of amylose and amylopectins. Okay, so first we will get amylodextrin, that will give violet color with iodine. Next we will get erythrodextrin, which will give red color with iodine, and this is mildly reducing. Next, acrodextrins, where there is no color with iodine. Okay, next comes maltose. What is maltose? Disaccharide containing alpha 1,4 linkage with glucose. Very good. So whether maltose is reducing or non-reducing? Reducing. So this will not give color with iodine, but it is very strongly reducing, and this will give positive which test? Benedict's test. So we detect the reducing property of any carbohydrate by Benedict's test. So maltose will give positive Benedict, whereas starch is negative for Benedict's. Next. Now come to glycogen. So what happens in glycogen? Glycogen is also the polymer of alpha D glucose molecules. But where is the difference from amylose and amylopectin and glycogen? Other than the source or the origin. These are plant origin, this is animal origin. Other than that, the branching of glycogen is more. Okay, I told you in amylopectin the branching occurs at every 20 to 25 number of glucose, whereas in glycogen it occurs at every 10 to 18 units. So the branches are dense. That helps in containing more glucose molecules. It is more compact than amylose and amylopectin, and where it is stored in animal liver and muscle. Okay, so the liver glycogen helps in storage of glucose in the form of glycogen in well-fed state, and in fasting state that glycogen helps in release of free glucose. Okay, whereas the muscle glycogen cannot be used as free glucose or cannot be passed on to other tissues, because why? Muscle lacks the enzyme glucose 6-phosphatase. That is why the last product of glycogen breakdown in muscle will be glucose 6-phosphate. Okay, I will show it later. Now, the glycogen is composed by glucose units joined by alpha 1,4 straight chain and alpha 1,6 branching points. Same as amylopectin, but the branching points are more frequent. Here, in liver, from glycogen, if we need glucose, we can convert glycogen into glucose, or we can break down the glucose molecules up to free glucose by various steps, and it is facilitated by the enzyme glucose 6-phosphatase, which helps in conversion of glucose 6-phosphate to glucose, and this glucose can come into blood circulation and go to any part of the body, whereas this glucose 6-phosphatase enzyme is not present in muscle. That is why the glycogen stored in muscle will be used by muscle itself, not by any other part or any other cells of body. Okay, so deficiency of this enzyme leads to a storage disorder, glycogen storage disorder. If our body is storing glycogen in a normal way, but it cannot break down glycogen to glucose for general utilization or in fasting state, if it cannot break down the glycogen, so because of the absence of this or inappropriate form of this enzyme, it will lead to storage of glycogen or increased store of glycogen, which will lead to hepatomegaly. What is hepatomegaly? Enlargement of liver because of excess glycogen stored in liver. Okay, so the disease known is this enzyme deficiency, it has a name. It is von Gierke's disease. Von Gierke's disease. Glycogen storage disorder, von Gierke's disease. It will come later on in your glycogen metabolism. Okay, just know that there may be a disease because of absence of glucose 6-phosphatase in the breakdown pathway of glycogen to glucose. Next. Now come to cellulose. Cellulose is a structural polysaccharide of plant. It is made up of glucose units combined by beta 1,4 linkage. Okay, so till now starch, glycogen, all we studied were in alpha 1,4 glycosidic linkage. Here comes the cellulose, which is beta 1,4 glycosidic bond. Okay, it is a straight linear structure with no branching point. Okay, so no branching point, it is a linear structure. Here, what is the point of or the importance of cellulose in our body? As humans, we lack the enzyme cellulase. Cellulase, what helps in the breakdown of beta 1,4 glycosidic bond. That's why we cannot digest cellulose. Okay. Next. So here is the structure of cellulose. So in a plant cell, the cell wall is composed of cellulose microfibers. Okay. And the individual cellulose microfiber looks like this. So it is a chain of beta 1,4 glucose polymer. Clear? Is this beta? Yes. Here it is. No, not alpha. The thing is rotated. The structure is rotated. Here it is 6 carbon. It is 4. Here it is 1 and again here it is 4. Okay. The alternate glucose molecule just rotates to make it continuous beta 1,4. Clear? It is beta 1,4 only. And here are the hydrogen bonds formed between the individual filaments of cellulose, making it compact and strong. The hydrogen bonds in between the parallel cellulose polymers make it compact and strong. Thus, the integrity of the plant cell wall is maintained. Okay. Clear? The structure of cellulose clear? Beta 1,4 glycosidic bond. Next. So what is the importance of cellulose in our body? Although we cannot digest it, what is the importance? It forms the major part of dietary fiber, which is non-digestible, and it can absorb up to 10 to 15 times of its weight in water. Okay, resulting in accumulation of water in the intestine. That helps in relieving the constipation and also by the increasing bowel motility. Next. This also helps in binding the bile salts with the fibers, which further helps in decreased fat absorption. We know that bile salts help in fat absorption in our gut. So without the bile salts, excess fat or excess triglycerides lipids cannot be absorbed. So it further helps in lowering serum cholesterol and triglyceride level. Next. It can also bind various toxic substances and eliminate them in the stool, thus decreasing chances of cancer, and also it delays gastric emptying. What is gastric emptying? It is different from peristalsis. Gastric emptying is emptying of stomach. Okay, so the more fiber or dietary fiber is there in our food, the easy satiety is achieved. Okay, so our stomach senses that the food is still in our stomach, okay, in our body, or we don't need more food right now. So it helps in reducing the glucose absorption also. Okay, the stomach releases the food, it will help in absorption of reduced glucose. Clear? So 4 main points I told you about the dietary fibers. One is dietary fibers or cellulose. First is retaining water and increasing the gut motility, resulting in relieving constipation. Second one is lower absorption of lipids or cholesterols, that is reducing the cholesterol level overall. Third is reducing the toxic substances and helps in reducing the chances of cancer. Okay, and the fourth point, the most important point, that is reducing the blood glucose absorption peak. Fine. Next. Here is a short note type of points. Dietary fibers come as your short note, so you can note this down. It is not absorbable in intestine as we lack the cellulase enzyme which breaks the beta linkage. And what are the examples of dietary fiber other than cellulose? That is hemicellulose, cellulose, pectin and lactulose. Lactulose I told you last day also, it is a synthetic disaccharide. They have high osmotic capacity to retain water and relieve constipation, increase bulk of stool and increase peristalsis activity, early satisfying of satiety by delaying gastric emptying. It is helpful in lowering blood cholesterol level and also has the anti-cancer role in colon cancer, and it is also helpful in weight loss. Why? It reduces the glucose peak in our body. Next is inulin. The next polysaccharide is inulin. What is inulin? Polymer of fructose linked by beta 1,2 linkage. Okay, it is found in, it is a reserve carbohydrate in bulbs and tubers like onion and garlic. Okay, it is used for renal clearance value and glomerular filtration rate because inulin is not absorbed, not digested and not secreted in our body. So, if we inject someone with inulin, the amount of inulin injected will be secreted by the urine, and it is different from insulin. Insulin is a hormone secreted by pancreas, which helps in lowering the blood glucose level in fed state, right? Next, dextrans. Now, we already studied about dextrins. What were dextrins? Partial hydrolyzed products of starch. This is dextran. It is branched homopolymers of glucose units connected by 1,6, 1,4 and 1,3 linkages found in microorganisms, okay, in bacteria, and it is not easily extravasated, so they are used for plasma volume expander as IV infusion. What is plasma volume expander? All the polymers, polysaccharides, they tend to collect water. Right? Because of the multiple OH groups, they all tend to accumulate water. So, if we inject this in someone's body, it will help in maintaining the blood volume by retaining enough water. ঠিক আছে, একটা এক্সাম্পল আমি দিয়েছিলাম, কারণ মনে আছে? ইন্ট্রাক্রেনিয়াল প্রেশার ইনক্রিজ হলে কোনো একটা ডেরিভেটিভকে আমরা ইউজ করি, mannitol. Very good. So what is mannitol? Alcohol of mannose. Very good. So these are used in treatment of hypovolemic shock. Hypo means less. Hypovolemic means less volume of blood. So to keep the volume stable, we use the dextrans as intravenous infusion. Clear? Next. Okay, so what is the difference between dextrose, dextrin and dextran? Dextrose is nothing but normal D-glucose. Okay, the D-glucose form is known as dextrose, which can also be used as drips in case of hypoglycemia in diabetic patients. In hypoglycemia, we can give dextrose, D-glucose, nothing but pure glucose, D-glucose. Dextrin, partially digested product of starch, and dextran is a high molecular weight carbohydrate or highly branched homopolysaccharide of glucose synthesized by bacteria. Clear? Next is chitin. Chitin is found in exoskeletons of insects which have the N-acetyl glucosamine with beta 1,4 glycosidic linkages. What is N-acetyl glucosamine? Amino sugar. Amino sugar means in which carbon of glucose amino group is attached? C2. C2. Here not only it is amine, it is acetylated amine, that is NHCOCH3. Okay, so the units of N-acetyl glucosamine is connected to each other by beta 1,4 glycosidic linkage, and this is found in insects. Next. Now come to heteropolysaccharides. Heteropolysaccharides can also be divided into plant and animal source. So plant heteropolysaccharides are hemicellulose, agar and pectin. Here are the sources. Plant cell, we found hemicellulose. In barks, we found pectin, and mostly in seaweeds, agar is found. Okay, here the linkage is beta 1,4 linkage. Various pentose sugars and hexose sugars are there. It is for part of dietary fiber. Now what is agar? It is a repeated unit of sulfated galactose and glucose. Okay, not only galactose and glucose but sulfated galactose and glucose, that also helps in retaining water. Okay, gel that helps in formation of gel-like structure of agar and agarose. Okay, what are the functions? We use agar in bacterial culture medium and electrophoresis in our lab. Okay, now pectin is also part of dietary fiber. Next, so agar is composed of linear polysaccharide agarose and agaropectin. Okay, so agar itself is composed of agarose and agaropectin. So agarose is a finer form collected from agarose. Okay, agar is composed of both agarose and agaropectin, whereas agarose itself dissolves in water, agaropectin doesn't. Okay, and it cannot be digested by bacteria, that is why we use it in bacterial colony culture. Now coming to mucopolysaccharides or glycosaminoglycan. This is the most important part of today's lecture, that is glycosaminoglycans. So you have to pay a little bit more attention in this part. Okay, this might seem very boring and difficult, but the questions, all the questions come from this part only. Majority question comes from this part. Okay, so glycosaminoglycans. Glycans mean polysaccharides. Glycans mane polysaccharides. Glycosamino mane carbohydrate containing amino groups. So what are the carbohydrates containing amino groups? Amino sugars. So it is glycosaminoglycans are nothing but repeating units of dimeric form of acidic sugar and amino sugar. Okay, here the glycosamino is made up of acid sugar and amino sugar. Glycosaminoglycans. Glycans means the polymer or the polysaccharide chain, and acid sugar, they can be of two types, glucuronic acid and iduronic acid. What is the difference between glucuronic and iduronic acid? The placement of COOH in the sixth carbon. If it is above the plane, it is known as glucuronic acid, it is D form. If this acid group is below the plane or within the ring, it is known as iduronic acid, L-iduronic acid. And the amino sugar can be glucosamine, galactosamine, N-acetyl glucosamine, N-acetyl galactosamine. Clear? So, amino sugars are of four types: simple glucosamine, simple galactosamine, N-acetylated glucosamine, N-acetylated galactosamine. Whereas the acid sugar can only be of two types. What are they? Glucuronic acid and iduronic acid. So polysaccharide, mucopolysaccharide name came from the word mucin, which was isolated from mucus. Okay, so that's why they are named as mucopolysaccharide. They are unbranched and with repeating disaccharide units of amino sugar and uronic acid. Uronic acid is nothing but sugar acids. Okay, so the amino sugars can be of glucosamine nature or galactosamine in nature, and the uronic acid is D-glucuronic acid or L-iduronic acid. ঠিক আছে? তো দুটোই জিনিস থাকবে, either অ্যাসিড থাকবে একটা আর একটা অ্যামিন থাকবে। তো অ্যাসিড ক্যান বি glucuronic or iduronic, and the amino sugar can be glucosamine or galactosamine. And they are joined by beta 1,3 or beta 1,4 glycosidic bond. Here, one amino sugar with uronic acid, they are connected by, if this is connected by beta 1,3, next one will be connected by beta 1,4, and thus the alternate bonding happens. Okay. And the amino sugars, the glucosamine or galactosamine, they remain in sulfated form. Okay. Next. Here are the structures of glucosamine. Can you appreciate the structure of glucose with the amino group at C2 point? C2. Taito? So this is simple glucosamine. This is glucuronic acid where the COOH group is above the ring. In iduronic acid, it is within the ring. Both are acids of glucose. This basic structure is similar to glucose, but it is the acid form of glucose known as glucuronic acid D form and iduronic acid L form. Why? Because this is the fifth carbon, penultimate carbon. D or L form is derived from the arrangement of OH group, position of OH group in penultimate carbon. For glucose it is C5. What is the difference between glucose and galactose? C4 epimer. So if this is galactosamine, what will be the change? In C4, OH will be above the plane. Okay. So the amino sugars, glucosamine or galactosamine. This is simple glucosamine or galactosamine. If this is N-acetylated glucosamine, তাহলে কি হতো এখানে NHCOCO3 CH3 will be there. Okay. In case of galactosamine, N-acetylated galactosamine, the OH will be above the plane and it will be NHCOCH3. Clear? Next. Now, what are the use of mucopolysaccharides or GAGs? Okay. So, this polymer of amino sugar and uronic acid, this is attached covalently to proteins called core protein to form proteoglycans. Now, these are the polymers of GAGs, or this is the mucopolysaccharides. They are attached to a core protein forming a bottle brush structure. Clear? Bottle brush, a brush used to clean the bottles. So, this structure resembles a structure of a bottle brush. Now, this core protein, this is further attached to a linking polysaccharide, linking mucopolysaccharide known as hyaluronic acid. Okay. So, in proteoglycans, the percentage of carbohydrates is up to 95%. Whereas, in last class, I told you in glycoproteins, the amount of carbohydrate was lesser than 10%. In glycoproteins, though the word glyco comes first, but it is majorly protein with few carbohydrates. But in proteoglycans, the protein part is less, whereas the mucopolysaccharide or the carbohydrate part can be up to 95% of the total weight of the proteoglycans. Next. Okay, so see this yellow line, this is the hyaluronic acid. Hyaluronic acid again itself a mucopolysaccharide, and various structures of proteoglycan monomers, they are connected with the hyaluronic acid backbone, forming the proteoglycan aggregates. So what is the role of this proteoglycan aggregates? They form the structural part of extracellular matrix in our body. Extracellular matrix mane ki? The portion outside each cell. But tissue is not only cell, tissue is made up of cell and extracellular matrix. The extracellular matrix part is made up of this proteoglycan aggregates with collagen and elastin and other microfiber proteins, right? So this proteoglycan aggregates, they form a major part in extracellular matrix. Next. And these mucopolysaccharides, they contain acid sugar and they contain amino sugars and they contain multiple OH groups. So because of the multiple OH group and few sulfur groups also, they contain sulfated amino sugars. So sulfur groups also, all these helps in accumulation of water with the proteoglycan structure. Okay, that's why it is surrounded by a shell of water molecule. Okay, the structure proteoglycan aggregates, they tend to accumulate water. So there is a cover of water molecule around the proteoglycan aggregate structures. Okay, now when they are compressed, when extra force is applied on them, what happens? The water molecules are removed. Okay, and it gives a cushioning effect. The moment the pressure is released, the water molecule again accumulates. So it gives a cushioning effect in the extracellular matrix. Next. So this is the thing. Here the mucopolysaccharides, they are surrounded by water molecules. When compressed, water molecules are released. Again, when the pressure is released, water molecules tend to aggregate. Okay. So these properties, they are seen in synovial fluid, vitreous humor, okay, and in the joint space, the mobility and the lubrication, they are maintained by this mucopolysaccharides. Next. Here are the few important glycosaminoglycans, their composition, the tissue distribution where they are found and their function. So, how to remember? How to remember the names of glycosaminoglycan? Okay, so another one is left, that is heparan sulfate. Heparan sulfate. So 6 glycosaminoglycans are there. Okay, another one is left, that is heparan sulfate. It is similar to heparin, but sulfur groups are more in number. Okay, and heparin is intracellular. Can you say what is the major function of heparin in our body? Anticoagulant. So it is mainly intracellular mucopolysaccharide found in mast cells. Okay, mast cells, and when released, they act as anticoagulant. Okay, so in all these glycosaminoglycans, see how many sulfates are there. Chondroitin sulfate, darmatan sulfate, keratan sulfate. Another is heparan sulfate. Okay, to decrease to eliminate the confusion, now remember CDK. Okay, the mnemonic should be CDKH3. CDKH3. GAGs are CDKH3. So C is for chondroitin sulfate, D for darmatan sulfate, K for keratan sulfate. Next come H3. H3, hyaluronic acid, heparin and heparan sulfate. Okay. So in case of chondroitin sulfate and darmatan sulfate, the sugar amine is of galactosamine type. First two, chondroitin sulfate and darmatan sulfate. Don't remember like this. Okay, write it down as I am saying it. CDKH3. CDKH3. So C is for chondroitin sulfate, D is for darmatan sulfate, K for keratan sulfate, H3 hyaluronic acid, heparin and heparan sulfate. Now C and D, they both have the amino sugar as galactosamine. Galactosamine. No other mucopolysaccharide contains galactosamine. Only these two, C and D. Okay. So first two, CDKH3, first two contains galactosamine, and the acid sugar in chondroitin sulfate, it is glucuronic acid. In darmatan sulfate, it is iduronic acid. How to remember? D for darmatan sulfate, iduronic acid containing D. Okay, so darmatan sulfate contains iduronic acid as acid sugar, whereas chondroitin sulfate contains glucuronic acid. Only two types of sugar acids are there, glucuronic and iduronic. Iduronic acid is there in darmatan sulfate. Easy to remember, iduronic, D for darmatan. Okay, now chondroitin sulfate, it contains glucuronic acid and acetyl N-acetyl D-galactosamine. Both contains N-acetyl D-galactosamine. Okay, not simple galactosamine, N-acetyl galactosamine. Where are they found? Chondroitin sulfate mainly found in bone, tendon, cartilage, heart valves and skin. Whereas darmatan sulfate is easy to remember, it is found in skin, derma. Okay, and what are their functions? Helps to maintain the structure and shape of tissues. Clear? Next comes keratan sulfate. So in keratan sulfate there is no acid sugar. In keratan sulfate, why it is placed just after C and D? Because they both contain galactosamine, whereas in keratan sulfate it contains normal galactose. Okay, so all three CDK, they contain something with galactose, CDK. So C and D, they contain galactosamine, whereas keratan contains simple galactose. No sugar, no acid sugar. Okay, no acid sugar. And the amino sugar here is N-acetyl glucosamine. So various MCQs can come from this part, stating that which mucopolysaccharide does not contain any acid sugar. Which one? Keratan sulfate. Keratan sulfate, no acid sugar is there. So where keratan sulfate is found? In cornea and tendons. So keratan sulfate of cornea keeps cornea transparent by accumulating enough water. Okay. Next comes the hyaluronic acid. Hyaluronic acid is the simplest GAG. It has D-glucuronic acid and N-acetyl D-glucosamine. Both glucose sugar and glucosamine in hyaluronic acid. Clear? In case of heparin, it is sulfated glucosamine and glucuronic acid or iduronic acid. Both can be there. In case of acid sugars, heparin and heparan sulfate, they contains both glucuronic acid and iduronic acid in alternate sequence, in random sequences. Okay. Glucuronic acid and iduronic acid both can be present, and the amino sugar is simple glucosamine with sulfur groups, sulfated glucosamine. So what is the difference between heparin and heparan sulfate? In heparan sulfate, the sulfur groups are more in number. Both found in these organs, but heparin is found in a special intracellular part, that is in mast cells, and heparin helps in anticoagulation also. Hyaluronic acid is found in all other connective tissues, synovial fluid, vitreous humor, and most importantly, the gel around ovum contains hyaluronic acid. Okay, the gel around ovum contains hyaluronic acid, and the main function is lubrication and shock absorbent in joints, hyaluronic acid. So the composition of hyaluronic acid is D-glucuronic acid, N-acetyl D-glucosamine. So how many mucopolysaccharides contain glucosamine? How many mucopolysaccharides contain glucosamine? Chatte. Other than C and D, সবকটাই আছে glucosamine. Simple glucosamine আছে heparin and heparan sulfate. Heparin and heparan sulfate, sulfated glucosamine. But in hyaluronic acid it is N-acetyl glucosamine. Also in keratan sulfate, N-acetyl glucosamine. Okay. Iduronic acid is there in darmatan sulfate and heparin and heparan sulfate. So glucuronic acid is exclusively present in chondroitin sulfate and hyaluronic acid. All the combinations can come as MCQs. Clear? Next. So this is the structure of hyaluronic acid. This is glucuronic acid. Is it clear? Glucuronic acid with N-acetyl glucosamine. Clear though? Detailed structure of hyaluronic acid. The repeating sequence of glucuronic acid and N-acetyl glucosamine connected by beta 1,3. Beta 1 and 3. Clear? But the next connection will be beta 1,4. Next connection. This is beta 1. This will connect with the 4 carbon of glucuronic acid. So the linkage is alternative. Beta 1,3, beta 1,4, beta 1,3, beta 1,4. Next. So this may come as a short note or any question explain why. Hyaluronidase. It is an enzyme that helps in breakage of beta 1,4 linkages of hyaluronic acid. And this is present in high concentration in seminal fluid and in few snake and insect venoms also. So the presence of hyaluronidase in seminal fluid helps in clearing the gel around the ovum, allowing the fertilization. Clear? So the gel around the ovum containing hyaluronic acid is broken down by hyaluronidase present in seminal fluid, helping in fertilization. And the hyaluronidase of bacteria helps their invasion into animal tissues also, as they are the connective tissue composition, hyaluronic acid. So the hyaluronidase from bacteria, they may also help in invading the cells of animals. Next. Now comes the chondroitin sulfate. কি ছিল chondroitin sulfate? Glucuronic acid, D-glucuronic acid and galactosamine, N-acetyl galactosamine. So galactosamine, sulfated galactosamine. Sulfur groups can be attached into C4 of galactose or C6 of galactose. Okay. They are also connected by beta 1,3 and beta 1,4. Beta 1,3 with the uronic acid with amino sugar and amino sugar to uronic acid is 1,4. So alternate bonds of beta 1,3 and beta 1,4. Can you appreciate this is glucuronic acid with N-acetyl galactosamine. Okay, so the chondroitin sulfate contains repeating units of glucuronic acid, beta 1,3 bond with N-acetyl galactosamine, and sulfur group can be present in 4 or 6. Okay, so this is chondroitin sulfate. Next, darmatan sulfate, iduronic acid. Iduronic acid, the structure of iduronic acid and N-acetyl galactosamine. Both C and D, they contain galactosamine, N-acetyl galactosamine. Chondroitin sulfate and darmatan sulfate. Galactose, N-acetyl galactosamine connected by beta 1,3. In chondroitin, it was glucuronic. In darmatan, it is iduronic. Okay, next. Keratan sulfate. Keratan sulfate only GAG not having uronic acid or acid sugar. So this is simple galactose, sulfated galactose. Galactose C4, this is not glucose, this is galactose connected by beta 1,4 with glucose N-acetyl glucosamine. Keratan sulfate is made up of galactose and N-acetyl glucosamine connected by beta 1,4. Clear? Next, this is heparin. Heparin, they can contain glucuronic acid and iduronic acid both with simple glucosamine, sulfated glucosamine. Glucosamine, not N-acetyl glucosamine, simple glucosamine, and they are connected by alpha 1,4. Alpha 1,4. Heparin is connected by alpha 1,4. So this can also come as a question. Which mucopolysaccharide contains alpha bond, alpha linkage? Which mucopolysaccharide contains alpha linkage? Which one? Heparin. Heparan sulfate also contains the same, where the sulfur groups are more in number. Glycosaminoglycans are generally extracellular, only heparin is intracellular. They are the component of mast cells and they act as anticoagulant. This also, heparin also helps in release of the enzyme lipoprotein lipase. Okay, lipoprotein lipase helps in breakdown of triglycerides into glycerol and free fatty acids. So the capillary wall contains the heparan sulfate with lipoprotein lipase. Heparin displaces this lipoprotein lipase, thus this can act on TG, and the heparan sulfate is present on the capillary endothelium. Okay, next. So these are the take home points. So hyaluronic acid, it is the linking glycosaminoglycan in proteoglycans. Okay, and the glycosaminoglycans, they are non-covalently linked to the hyaluronic acid, whereas the hyaluronic acid contains no sulfate group. This can also come as a question. Hyaluronic acid, there is no sulfate group, whereas all other, they contain sulfate groups. Chondroitin sulfate, darmatan sulfate, keratan sulfate, heparin and heparan sulfate. All other 5, they contain sulfate groups. Okay, glycosaminoglycan with no uronic acid is keratan sulfate. What is their? Simple galactose. And they are mainly extracellular, exception is heparin. Next. So now, diseases related to mucopolysaccharides is known as mucopolysaccharidoses. Okay, mucopolysaccharidoses. They are mainly autosomal recessive. What is autosomal recessive? Cell mediated, cell linked, not gender specific, except Hunter syndrome, that is X linked. What is the criteria or what is the result of X linked diseases? X linked diseases are seen in case of for females it should be homozygous. Okay, in case of males, so it is obvious. Okay, so for males, X-linked diseases are more prominent, like they always show the disease. Only one X is there, so it will show the disease. Biochemical defects in mucopolysaccharides, what happens? The degradation of mucopolysaccharides by lysosomal hydrolase is impaired. So the enzyme lysosomal hydrolase, if this is impaired or absent, this lead to impaired degradation of mucopolysaccharides and this leads to storage of abnormal mucopolysaccharides in our body. Okay, so the lysosomal vesicles, they become swollen with incompletely degraded GAGs. So now the GAGs half-lives are 3 to 10 days except keratan sulfate, that is 120 days. So what happens? The impaired degradation leads to rapid accumulation within the cells. Okay, and this can be accumulated in liver, spleen, bone, skin and CNS. Everywhere where the GAGs are present. Next. The clinical symptoms will be mental retardation, coarse facial features, corneal clouding. Which GAG helps in corneal transparency? Keratan sulfate. Okay. Visceromegaly. Visceromegaly means enlargement of viscera. Short stature, that is reduced height or decreased growth of bones. Joint contractures because of loss of lubrication and synovial fluids. Okay. Now how to diagnose this? The excessive secretion of GAGs in urine can be found, or prenatal diagnosis. What is prenatal diagnosis? Before birth. Okay, so before birth diagnosis is possible by amniocentesis. Amniocentesis means amniotic fluid extraction, can help in diagnosis of mucopolysaccharidosis by estimation of lysosomal hydrolase enzyme concentration. Okay, next. These are the 7 types of mucopolysaccharidoses. Type 1, Hurlers. The enzyme defect is L-iduronidase. Okay. Next, Hunters. Here it is also iduronate sulfatase. Next, third is Sanfilipose, heparan sulfatase. Fourth is Morcus. It is galactosamine sulfatase. Fifth is Kais, where it is also L-iduronidase. In 6, it is Martix Lamy. The enzyme defective here it is N-acetyl galactosamine 4-sulfatase. So all these enzymes help in formation of the components or digestion of the component. Okay, so any defect in these enzymes lead to the faulty production of GAGs or accumulation of impaired products. And the 7th one is slice, where beta glucuronidase is defective. Okay, accumulated in these particular diseases. Among all these diseases, all of them are autosomal recessive, not dominant, autosomal recessive. But Hunter's is only X-linked. Now answer the questions. Which of the following polysaccharides serves as the primary storage form of carbohydrates in animals? What is the answer of question 1? Taratari, shomoy nei. Which of the following polysaccharides serves as the primary storage form of carbohydrates in animals? Jore bol. Jore bol. Glycogen, right? Okay, glycosidic linkage in cellulose is beta 1,4. Taito? Beta 1,4 to. Next. Which polysaccharide forms the major structural component of fungal cell walls? Fungal cell walls. Fungal cell wall. Not insects. Chitin. Tick. Tick to. Okay. Branching in glycogen occurs through which type of glycosidic bond? Branching. Alpha 1,6. Next. Which of the following statements about cellulose is false? Which statement is false? It is composed of beta D glucose unit, forms microfibrous by hydrogen bonding, it is digestible by human enzyme, provides dietary fiber. C. It is not digestible by human enzymes. Which test can detect reducing polysaccharides? B. Benedict's. Next. Hyaluronic acid differs from other glycosaminoglycans by not being sulfated, having fucose residues, being branched, containing only hexoses. Not being sulfated. Heparin is highly sulfated polysaccharide mainly composed of. Heparin is mainly composed of N-acetyl glucosamine and galactose, glucuronic and iduronic with glucosamine, mannose and fructose, galactose and fucose. Easy. A. B. Heparin contains both glucuronic and iduronic. Heparin and heparan sulfate contains both the acids, glucuronic and iduronic acid with glucosamine. Next. A group of inherited lysosomal storage disorders caused by a deficiency of specific lysosomal enzymes is the definition of Mucopolysaccharidosis. Okay. Next, which type of MPS is an X-linked recessive disorder? Hurler, Sanfilippo, Hunter, Marquis. Hunter. How to remember? X-linked, Hunter. Mostly Hunters are male. So X-linked disease is Hunter's disease. Okay? Okay.